Hello students, today our topic of study is the fluid mosaic model of cell membrane, which is also called as plasma membrane or plasma lemma. The fluid mosaic model of cell membrane, plasma membrane or plasma lemma. Okay, now suppose this is a this is an animal cell and it is its nucleus and this is cytoplasm. So it is cell membrane for an animal cell. Now what is cell membrane for an animal cell? Cell membrane is the outer lining which differentiates the cytoplasm from the outside of the cell is known as cell membrane for an animal cell. But if we consider a plant cell or a bacterial cell, their present cell wall, another boundary develops outside of cell membrane that is called cell wall in case of plant cell or animal cell. Plant cell or bacterial cell. So in case of plant cell or bacterial cell, cell membrane remains inside of the cell wall. But in case of animal cell membrane is the most outer lining of the cell that differentiates cytoplasm from outside of the cell. Now some general characteristics of cell membrane. Cell membrane is semi-permeable or selectively permeable in nature. Selectively permeable in nature means cell membrane is permeable to all solvent, but it is not permeable to all solutes. Some solutes can get through it, can be diffused through it, but some could not get diffused through it. So it is semi-permeable in nature, or it is also called as selectively permeable in nature. Its thickness remains within 70 to 100 angstrom. Now for your idea, 1 angstrom is 10 to the power minus 10 meter. So you can easily imagine the actual thickness of a cell membrane, and its main components is lipid, which remains around 80%, proteins, which remain around 20%, and a rare percentage of carbohydrate also present in cell membrane. In lipid, mainly phospholipid, cholesterol, this is present in cell membrane. In case of protein, structural protein, carrier protein, this is present in cell membrane. And carbohydrate, glucose molecule, this is present in cell membrane in rare percentage. Now, if we come to our main topic, which is Singer, which is fluid mosaic model. So, at first, we need to know when electron microscope invented in the world. So electron microscope invented in the year 1958. And after invention of electron microscope, the most accepted model which can most convincingly explains or describes the structure and function of a cell membrane was fluid mosaic model. Fluid mosaic model, which came around 1979 AD, which is proposed by Singer and Nicholson. Now, we need to draw a general diagram of this model to get a better understanding of this model. Now, this is the polar height of the lipid molecule, and this is the non-polar tail of a lipid molecule. Here, this is also polar height, remain in opposite direction, and this is the non-polar tail. This is the integral or intrinsic protein. This is also polar height and non-polar tail, polar height and non-polar tail of lipid molecule. Now, this is some part of extrinsic protein. Here extrinsic protein, this is glucose molecule. This is also glucose molecule. This is glucose molecule, and this is also glucose molecule. So, this is extrinsic. This is extrinsic or peripheral protein. Peripheral protein. This is extrinsic or peripheral protein. This is intrinsic or integral protein molecule. This is carbohydrate or glucose molecule. Glucose molecule. And this is that height part of a lipid molecule which is polar in nature, which is also hydrophilic. And this is the tail part of a lipid molecule which non-polar in nature and which is also hydrophobic in nature. So it is a general structure of Singer-Nicholson model in a diagrammatical view. So before coming to Singer-Nicholson model or fluid mosaic model, we need to discuss about another model. So after invention of electron microscope in 1959 AD, there came another model which is Robertson trilaminar model. Trilaminar model. So this model, Robertson trilaminar model, came before of fluid mosaic model proposed by Singer-Nicholson. The Robertson trilaminar model came before of fluid mosaic model in around 1959 AD, which postulates that presence of lipid bilayer in a cell membrane and presence of two protein layers covering the lipid layer. Covering the lipid layer. So, presence of lipid bilayer already postulated by Robertson trilaminar model before of this fluid mosaic model. So, concept of lipid bilayer came before of fluid mosaic model by Robertson trilaminar model. So, Robertson trilaminar model gave a general structural orientation of the cell membrane, and trilaminar model also postulated that presence of two protein layers covering the lipid layers or covering the lipid bilayer. So the basic structural orientation for Robertson trilaminar model was that this type, means this is a protein layer, this is a protein layer, it is outside protein layer and it is inside protein layer, and in between them the lipid bilayer get sandwiched. Okay, so it is the lipid bilayer and it is the outer covering of protein layer, and it is the inside covering of the protein layer, and this protein layer thickness remains around 20 angstrom and the lipid bilayer thickness remains around 35 angstrom, and it is also 20 angstrom. So altogether it results 75 angstrom in thickness, which is more similar of thickness remains between remains within 70 to 100 angstrom. So this was postulated by Robertson trilaminar model before the fluid mosaic model proposed by Singer-Nicholson. But this model could not convincingly describe the all functions of plasma membrane. Now we are coming to fluid mosaic model. And what is the differences between this Robertson trilaminar model and Singer-Nicholson's fluid mosaic model? So now you follow my words very attentively. If we look at the diagrammatical view of Singer-Nicholson's fluid mosaic model, we can see this is the head part of the lipid molecule or polar part of the lipid molecule, which is hydrophilic in nature, and this is the tail part of the lipid molecule. So this is the first layer. This is the first layer of lipid molecule or lipid layer, and this is the second layer of the lipid molecule or lipid layer. So this is from the lipid bilayer. Okay, so here the head remains at the outside direction, and here the head remains at the inside direction of the cell membrane. So they remains at the opposite direction. So inside of the cell membrane, inside part of the cell membrane remains polar in nature, remains here the polar head of the lipid molecule arranged, and here the in the outer side outer part of the cell membrane, the polar part of the lipid molecule arranged. Okay, now if we look at the extrinsic protein part, here we can see that the extrinsic protein not remain in continuous from they remained at uh, disorganized from here and there, here and there. But here in Robertson trilaminar model, it postulated that the two protein layer covering the lipid layer, two protein layer means in continuous from, covering the lipid layer in continuous from. But if we see the diagrammatical view of Singer-Nicholson model, we can clearly see that the protein molecules remained in random orientation, not in continuous orientation. here and there in disorganized manner. Okay, and the integral protein remained embedded in the lipid molecule, but there is no mention of embedded protein molecule part in Robertson trilaminar model. So here is the basic differences of Robertson trilaminar model and fluid mosaic model. And the integral protein remained closely attached with the lipid molecule, or tightly attached with the lipid molecule, because it remained embedded in the lipid bilayer. But the extrinsic protein remain in surfacing condition above the lipid layer. Okay. Now, one more thing to add here, in fluid mosaic model, Singer Nicholson proposed that the lipid bilayer form remains in fluid form and constant motion form, and they also undergo lateral diffusion. But and as the lipid layer undergo lateral diffusion, the protein part also move rapidly from here to there. And if we remove the extrinsic protein part, the, the main structure of the cell membrane cannot get disrupted or changed. But if we remove the integral protein from the lipid bilayer or from the cell membrane, the total structure of cell membrane get disrupted or violated. Okay, and it is the carbohydrate molecule get attached with the extrinsic or intrinsic protein and also the lipid molecule. So this type of protein molecule which get attached with the carbohydrate molecule is known as glycoprotein, and this type of lipid molecule which get attached with the carbohydrate molecule is known as the glycolipid. Now one more thing to add here, one concept, one idea, or one term arise from this model, from this Singer-Nicholson model, that is glycocalyx. And glycocalyx is formed by the carbohydrate covering. Carbohydrate covering means the glycolipid and the glycoprotein part of extrinsic and intrinsic protein, and the glycolipid part, which form a carbohydrate covering around the cell, and which is known as glycocalyx. And the main function of glycocalyx to recognize the foreign protein and to protect the cell from the outside digestive juice, which can digest the cell also. So it is the structure of glycocalyx and it is the function of glycocalyx. Now, if we come to the function of cell membrane, the basic function and the prime function of cell membrane is the transport of material from inside of the cell to the outside and outside of the cell to inside. Transport the molecules, and it is the basic and the most prime function of the cell membrane. And it can be done in two modes, which is passive transport. In case of passive transport, no energy is required. Okay, no energy is required, and it can be done in two ways. First one is simple diffusion. Simple diffusion occurs just from high concentration to low concentration, maintaining the concentration gradient. Okay, and number two is facilitated diffusion. Facilitated diffusion, it also occurs from high concentration to low concentration, but here a carrier protein is required. Carrier protein is required to to occur the to occur this facilitated diffusion. Mainly some polar molecule cannot get through this non-polar part. Here it is obstructed, cannot get through this non-polar part of the lipid molecule, and here a carrier molecule requires to do the facilitated diffusion, maintaining the concentration gradient, okay, from high concentration to low concentration also. And number two is number two the second mode is active transport. Active transport, here energy is required. Here mainly the active transport occurs against against the concentration against the concentration gradient, means from low concentration to high concentration. So here energy requires, and it is also happened in two from. One is primary active transport, where direct energy consumption is required, and two is secondary active transport, where indirect energy consumption is required. So this is all for today's topic, and here we describe the fluid mosaic model of cell membrane. So thank you, students.